Can you put a timer for me?
Yeah, well, I put the microphone. Good morning, ladies and gentlemen. Thank you for attending this press conference about the warning of more than 100 scientists and policy experts on the danger of genetic biotechnologies, on pollinators and biodiversity. My name is Vanessa Mermet from French NGO Pollinis. We work on pollinator protection and initiated the appeal to protect insect pollinators from the potential negative effects of genetic biotechnologies. This text has been signed by more than 100 scientists and policy experts from 30 countries in all continents. They warn on the additional dangers that a release in natural habitats of genetic biotechnologies such as RNA pesticides or gene-drive organisms, could have on insect pollinators, potentially precipitating their ongoing extinction. To date, limited research has been conducted to understand the risk and impact on pollinators of such a release. It is not possible to provide robust and reliable risk assessments to ensure that pollinators' decline will not be further precipitated by the release of these technologies. Therefore, the signatories of this appeal call world leaders gathered here at COP for a strict application of the UN precautionary principle. They call upon the parties and signatories to the UN Convention on Biological Diversity to oppose the deployment in nature of organisms, products, and components from genetic biotechnologies at international, regional, and national levels. The speakers here today are signatories of this appeal, either as individuals or as organizations. They will present you with the issues related to the development of genetic biotechnologies and diversity, and what are the real solutions we should put in place in order to really protect biodiversity and no further threaten or destroy it. First, I would give floor to Professor Lucas Garibaldi. He's a renowned scientist whose research work is aimed at promoting biodiversity, a healthy food production system, and physical, mental, and social well-being. He's interested in agroecology, pollination, and the contributions of nature to society. He's co-chair of IPBS and coordinates important working groups, including INLAD, Beetles Agroecology in Argentina, and beekeepers associations, including Apimandia. Please.
Thank you. Okay. Hello, everybody. So we have many speakers here. I will try to be fast, and I will focus on the agricultural sector to complement my colleagues here, and especially on the ecological part. So it is thought that these new biotechnologies can be a solution for the conservation of biodiversity. And in my opinion, this is completely wrong, because these technologies are being developed by big companies within one type of production system, which is called industrial agriculture. So industrial agriculture, or what we call land use change, is the first driver of biodiversity loss. So these new biotechnologies are designed to work within these systems of large monocultures that do not promote biodiversity. On the contrary, these systems replace biodiversity with monocultures. So by no way developing technologies that work only under monocultures will help biodiversity. This is a contradiction at all. So importantly, I want to stress that nowadays we have other ways to produce healthy food and enough amount of food to feed everybody, which is food produced based on practices that are based on biodiversity. And there are a lot of examples from science and from practitioners from agroecology all over the world that there are other ways that food can be produced without the use of pesticides, or even without the use of these new biotechnologies that are designed to work under these large monocultures. So thank you.
Thank you, Professor Garibaldi. I will let now you dig into molecular biology and the newest developments and risk of genetic engineering for biodiversity with Dr. Ricarda Steinberger. She's a molecular geneticist and developmental biologist specialized in synthetic biology, gene regulation, and genetically modified organisms and their risk and potential consequences on Earth, food security, agriculture, biodiversity, and ecosystems. She's presently co-director of Econexus. She has been involved in the process of CBD for a long time. And she's also a founding member of the European Network of Scientists for Social and Environmental Responsibility. Please.
Thank you very much. It's quite clear that, and we've heard that pollinators, I mean, all of biodiversity is under extreme stress in the moment. And pollinators, of course, are at the receiving end of a lot of our technologies and practices in terms of agriculture. There is now the idea of how can we counteract. And one would, for example, be like with the varroa mite, to use something like a gene drive. You might have heard of gene drives. That is a genetic technology that is specifically designed to override the normal inheritance rules, basically to drive a certain trait, a gene, into the future in the wild. So once released, it will keep spreading. I mean, we heard it from mosquitoes, but if you look, we've done a study that shows that gene drives are suggested for 32 different insects. And out of these, 21 actually are intended for agricultural pests. So you see the focus is on trying to keep the agricultural system going as it is. And the varroa mite, genetic modification, and trying to turn it into a gene drive, we don't know what the consequences may be, because we don't know how to assess gene drives. It's a very risky technology. The other area, which is interesting to look at and quite alarming, is the use of gut bacteria. I mean, we know like bees and pollinators will have gut bacteria. Every organism has, we have plenty. So if you modify the gut bacteria, for example, here it is suggested to make gut bacteria produce something like double-stranded RNA. What is that and why is it? It will interact with the gene expression. It will link to the DNA itself and stop the gene expression of certain genes. For that, you need to be quite specific which gene you want to block off. But of course, genes across all the species will have some familiarities. So it might be meant specifically, but there will be a lot of target impacts as well. And yet it's being strongly pushed as one of the new ideas to modify gut bacteria, to put them into the bees, and to have basically a defense system from inside, or to give the gut bacteria different enzymes so that, for example, they can help the bee digest pesticides. Of course, gut bacteria will go to other insects, will go to other species. It might actually also impact their own way of working if we modify them. What will they do? They might actually end up being a real problem, and how do we get them out? So you can see there is a lot of thinking to be done in terms of risk assessment, but with the new technologies, we really need here at the convention, for example, to make clear we have it on the horizon scanning, we have it in technology assessment, and in terms of the application, we should not let it go forward at present, because we do not know how to understand or assess the risks. Thank you.
Thank you, Dr. Steinbrecher. I will now give the floor to Dana Pearce from the organization Friend of the Earth USA. She will tell us about the risks and concerns of genetically engineered microorganisms in agriculture. So Dana is the Senior Program Manager for Emerging Technologies with Friend of the Earth and leads the food and agriculture teams, international and national regulatory and market campaigns on biotechnology and genetic engineering. Please.
Thank you so much. I'm going to elaborate a little bit from what we just heard from Dr. Steinbrecher and speak to a number of other new and emerging genetic technologies which may pose risky impacts to pollinators. One of those includes gene silencing sprays using an RNAi technology, which pose novel risks to pollinators, health, and the environment, and also raise concerning issues around ownership and patenting. Pesticide companies, including Bayer, BASF, and Syngenta, are using genetic engineering technologies to design new pesticide sprays that exploit a cellular process called RNA interference, RNAi. This is also known here as transient modification, and it's used in order to turn on or off, or in other words, silence genes that are essential for the survival of insects. And so if you use this RNAi and spray it, the RNAi will turn off the survival genes, thus trying to kill the insect. For example, there was a meta-analysis in 2017 that found that the interfering RNA could directly impact the genetic activity in honeybees. Another study in 2019 showed the uptake and exchange of interfering RNAs within bee colonies. So this genetic spray, gene silencing spray, which is used on crops, or the desire that they have is to use it on crops and to genetically modify a crop perhaps halfway through its life, is actually being taken up by pollinators such as honeybees, and also impacting and altering honeybee functioning in ways that we don't fully understand. This is not actually hypothetical. The first gene silencing pesticide spray, which is an RNAi spray applied to crops with the intent of killing the Colorado potato beetle, was submitted to the US Environmental Protection Agency for environmental release. Currently, it's under review. And once this is released into the environment, it could set off a chain reaction of gene alteration, in particular for honeybees and other pollinators. And this is a technology that cannot be recalled once that environmental release happens. The other risky new genetic engineering technology is genetically engineered soil microbes. Although this is not yet on the market, we are seeing a number of pesticide companies put a lot of resources into the development of genetically engineered soil microbes for use in agriculture. The idea is to release these genetically engineered microbes onto millions of acres of farmland. And this really opens up a new chapter in agricultural biotechnology. We're really only beginning to scratch the surface of our understanding of the complexity of microbial communities, from the microbiome to soil health to soil microbes, including the impact on pollinators. So these GE soil microbes are live organisms which reproduce and can interact with other species. These soil microbes in agriculture would represent an unprecedented level of open air experiment that could have irreversible impacts. So for example, we're talking about 3 trillion genetically engineered microorganisms for every half acre. This is coming from some of the research that's happening. It's really critical that we are not releasing these GE soil microbes or the RNAi gene silencing pesticides at this moment. We need real assessments, we need precautionary regulations before we even consider the environmental release into the environment. Thank you.
Thank you, Mrs. Peirce. Now we will hear Adam Brisley from the organization Save Our Seeds, who will give us field insights on the impact of biodiversity and livelihood on an agricultural application of gene drives. Adam is a human rights activist who has worked for 2 decades with communities around the world impacted by genetic technologies, and he has been involved in an interview project on gene drive. He will focus on the African, Uruguay, and Brazil example of new genetic technologies impacts.
Thank you. So the Gene Drive project you'll hear most about here is Target Malaria, which is a Gates Foundation project at Imperial College London that's particularly active in Burkina Faso, but is also active in many African countries and actively lobbying the African Union with the claim that they'll make mosquitoes disappear from the environment. There are many biosafety and ecology questions surrounding that project, including that mosquitoes and mosquito larvae are part of the food web, bats and fish, for example, or whether it would actually work to control malaria. But we should also not just think that gene drives are about mosquitoes. This is also part of Gates's push, such as Bill Gates's recent visit to Kenya, to open up Africa to GMO agriculture. In Kenya, Gates falsely claimed that all the bread he's ever eaten has come from GMO wheat, despite the fact that GMO wheat has only ever been recently approved to grow in very few countries. And we know that the expansion of GM agriculture is associated with the increase in agrotoxins that threaten pollinators. In July, I was at a target malaria conference in Greece, and there's a startup company called Biocentis that's housed in target malaria's facilities and insectaries in Italy and the UK that seeks to apply and sell the target malaria intellectual property to apply that gene drive technology to many other organisms as a kind of genetic pesticide. Another example is Uruguay, where the Institute Pasteur is developing gene drives in a fly pest that affects cattle. At the same time, the Uruguayan government is following the example of many countries and discussing removing the need for new genetic technologies to be subjected to regulatory oversight and risk assessment as GMOs. Last night here at the COP, Brazil's delegation was actively trying to block COP oversight of synthetic biology. Brazil was arguing that this technology is something in the future and so that it can be postponed to future discussions of the Biodiversity Convention. However, they are being very disingenuous because Brazil in 2008 became the first country in the world to pass legislation paving the way for the environmental release of gene drive organisms and also removing these genetic technologies from the need for regulatory oversight and risk assessment as GMOs under Brazil's law. Meanwhile, Brazil have allowed themselves to become the international dumping ground for pesticides and also a gateway to be exploited as a place to test these risky new genetic technologies and organisms. Post Dilma and under Bolsonaro, Brazil has approved around 1,500 new pesticides, many of which are banned in other countries. The permitted glyphosate levels in Brazil's drinking water are 5,000 times those allowed in Europe. Brazil is also a dumping ground for the technologies we're talking about here because they've removed the need for regulatory oversight. Brazil has commercially released GM mosquitoes, which have crossed and persisted in the environment and about which nothing is known, including the transgenic mosquito's capacity for disease transmission or their impact on other organisms. And they are also field testing the Oxitec company's GM fall armyworm, which is a Lepidoptera species. Brazil is a stark example because we're here at a meeting talking about saving biodiversity while one of the largest reservoirs of biodiversity in the world, the Amazon, is being burnt and cut down in large part to expand transgenic soya plantations. So all that Amazonian life is being destroyed with pesticide and herbicides and decimated. The GM soya is being exported to feed meat animals in Europe and also for the COP president China as the largest purchaser. So this expansion of green deserts into the Amazon is really something that should be on the table here at the COP. Thank you.
Thank you, Mr. Brisley. I give the floor to Dr. Tom Wakeford from ETC Group, who will present risk assessment issues. And Dr. Wakeford is a UK-based biologist and participatory action researcher at ETC. He holds honorary positions at the universities of Cambridge and Exeter and has used participatory approaches to technology assessment in Europe, South Asia, and Latin America. Please.
Thank you very much. Welcome, everybody. Yes, I'm from the Etc. group, and we monitor developments in this and other areas of emerging technology. Jennifer Doudna was presented with the Nobel Prize for inventing this technique. Many of you know, CRISPR-Cas9. She's undoubtedly clever, and she made a prediction a few years ago that actually the biggest application would be for CRISPR-Cas9 and gene drives would be agriculture. And you've heard from Ricarda Steinbrecher that this has come true. This is exactly how things are moving, and Dana Pills gave some of the other applications sort of in the field. Now, Jennifer Doudna may be clever, but I guess the question is she wise? Is this a wise technology to use in the fields of often agroecological settings, smallholder farmers, indigenous peoples who are the custodians of our biodiversity. What Jennifer Doudna was doing was kind of scanning the horizon. She was sitting there, I think before she'd even been given her Nobel Prize, and looking ahead. Now, scanning the horizon of the future is exactly what we should be doing. But what's emerging here at the CBD is that more and more people are realizing that that should be done systematically, not just by one clever person, but by people of all different perspectives, different scientists, who draw on different sorts of knowledge that may challenge that's just a scientist in the lab creating technologies that could have consequences they can't foresee. So this idea of horizon scanning and then assessing technologies that have emerged, and gene drives is the one that gets the headlines, but there are, you've heard others touched on here, and one that's just emerging, and you can hear about it, I think, a side event from the German Nature Conservation Agency at lunchtime is GM viruses and so-called self-spreading vaccines, which are, as we've learnt, I think you'll hear more about, actually being released in the United States at the moment with almost no regulatory oversight. That's exactly the sort of thing that technology assessment should be there to prevent. It's extremely dangerous. So we have proposals for a multidisciplinary ad hoc technical expert group here. We really need more of that applied to the whole area of synthetic biology. Goes without saying, pollinators are in the front line. And then for those technologies that have been released, that was such a dramatic example of glyphosate from Brazil. They need monitoring both for human and environmental health. So we call for scanning that horizon, doing technology assessment, all that in a participatory way, and monitoring the technologies as well. Thank you.
Thank you, Dr. Wackford. To conclude, I will give the final word to Nicolas Larman, Delegate General of Pollinis, who will give us the reasons why NGOs are calling for a moratorium. Please.
Bonjour à tous, je vais enchaîner en français, excusez-moi, normalement.
Good morning, everyone. I'm going to speak in French since interpreting is available. So, the word moratorium is being brought forward by Pollinis and its partners. And I'm going to give you a concrete example. Interfering aerial organisms. So, by targeting insects, these genetic pesticides which are developed will have the same catastrophic impacts on biodiversity that we've seen in the past, and which is one of the main issues threatening insects around the world. Genetic pesticides have the aim to reduce or destroy insects, but they can't make a difference between the good and the bad insects. Of course, the manufacturing companies pretend that they know how to identify the good and the bad, but we need to understand that these products target the different species, and there's no discrimination. So pesticides which deactivate some of the genes is a big problem. So we need to look at the effects. These are alarming effects which are provoked by these pesticides, and pesticides which are not always authorized. I remind you this. There are pesticides which targets the IE morphalis in Latin, was part of the publication 2008 of Reserve Nature, and this can potentially deactivate a vital gene in different species, 60% of wild bees and 11 bumblebees, and 4,200 of different species which are important for pollinization. In front of these risks, releasing organisms in nature from biotechnology is not a possible action. We therefore ask an urgent setting up of a moratorium for these pesticides and all organisms from which are genetically modified. We need to look at the conservation of different species in nature. We have other proven methods which are more natural to control and also to manage biodiversity, and will also give rise to more respect to the environment. These methods are very well documented nowadays, and there are no risks to the environment or organisms. We need to encourage these methods and support them by the different international institutions and the public. Thank you.
Of time, we have to leave the room for the next press conference. I will take only one question, and then if you have other questions, please ask to the speakers just next to the room, please. Okay, well then, thank you very much. Huh? Okay, no, I didn't see you, please.
Pour Nicolas, tu pourrais nous dire qu'est-ce que...
Or Nicholas, what are you expecting from COP15? What would you include, and in what of the objectives would you include this? What would be your recommendation to give us what you'd like to see happening? So I need to. There is someone else who's been following all the negotiations, and this person could be able to answer much better than I. So.
I think he wants us to go off that way, right? We normally go off that way, it doesn't matter.